Welcome to today's topic within hematology, hemolytic anemia due to inherited membrane and enzyme defects. We'll delve into the intricacies of this condition, exploring the genetic factors that cause the red blood cells in some individuals to break down prematurely, leading to anemia. Let's begin with the structure of the normal red blood cell. Picture this: the cell membrane is not just a simple boundary. It's a complex, highly organized structure composed of a lipid bilayer, integral and peripheral proteins, with an underlying skeleton of proteins like alpha and beta spectrin. This structure ensures flexibility and durability as red blood cells traverse the narrow and winding pathways of your circulatory system. Now, imagine what happens when this finely tuned system is disrupted. We see this in conditions like hereditary spherocytosis and hereditary elliptocytosis. This condition is most common among individuals of European descent. Typically inherited in an autosomal dominant pattern, its severity can vary widely. It could present at birth, later in life, or be discovered incidentally. The defect usually lies in one of the membrane proteins like ankyrin, band 3, or beta spectrin. Over time, affected red cells lose parts of their membrane, especially while passing through the spleen, turning more spherical and losing their flexibility. These cells are then prematurely destroyed, mainly in the spleen. Patients typically have symptoms of hemolytic anemia, and their spleens are usually enlarged. On a blood film, one would see microspherocytes and signs of polychromasia. Tests for hemolytic anemia are generally positive, and the eosin 5 maleimide (EMA) test for band 3 abnormality is particularly telling. Treatment often includes a splenectomy to improve red cell lifespan, though spherocytosis itself persists. In mild cases, surgery might be delayed, especially in children under 6. Prophylactic folic acid is recommended for severe cases, and attention must be paid to possible complications like pigment gallstones. Shifting our focus geographically and genetically, we find Southeast Asian ovulocytosis, another inherited defect of the red cell membrane, particularly band 3. Interestingly, carriers of this condition have some resistance to malaria, a brilliant example of how genetic traits can provide unexpected advantages against environmental challenges. Let's pivot to enzyme defects, starting with glucose 6-phosphate dehydrogenase, or G6PD deficiency. This condition affects the hexose monophosphate pathway and is crucial for protecting the red cell from oxidative damage. Located on the X chromosome, this disorder is more prevalent in males but can affect females with mild symptoms. G6PD deficiency leads to vulnerability in red blood cells under oxidative stress, such as exposure to certain drugs, infections, or even eating fava beans. This can trigger acute hemolytic episodes. Typically, the blood count is normal until a crisis hits. During an acute episode, features of intravascular hemolysis appear, potentially leading to renal failure in severe cases. Blood films may show bite cells and blister cells, with Heinz bodies visible upon special staining. Another enzyme defect is pyruvate kinase deficiency, which affects the glycolytic pathway and is the most common cause of chronic nonspherocytic hemolytic anemia. It's an autosomal recessive disorder where affected individuals may have symptoms that are mild compared to the degree of anemia. Treatment may include splenectomy to improve symptoms. To wrap up, inherited defects in red blood cell membranes and enzymes present a complex interplay of genetic, biochemical, and clinical challenges. Understanding these can help manage and sometimes even prevent severe complications in affected individuals. Thank you for tuning into this session. We will continue this topic with some acquired forms of hemolytic anemia in the next session.